Hello everyone, we're talking about carbohydrates again. As we did the previous day of lecture, we talked about monosaccharides and sort of the properties and how to distinguish those. And we also talked about a review of hemiacetals. And then in class today, we talked about making hemiacetals through closing the ring. So this bottom structure here on the right, that's the closed form of the sugars. So we talked about how to do that process. And then we also started talking about linkages. So I just want to pick up there and finish up this section so we can talk about disaccharides and polysaccharides. So let's get started with that. So just as a reminder about what we talked about in class, we have 4 different types of possible linkages. We have one for alpha, one for beta, 1,2 alpha beta, and 1,6 alpha. So all of those different types of bonds were shown on this slide, which we did in class, but just as a reminder, when you're either trying to draw or to recognize a different type of linkage, the number part, the 1,4 or the 1,6 or the whatever it is going to represent the number of the carbons on the glucose or the sugar that you have. So if it's the closed form here, if you look on the left, it's starting with carbon 1 on the left sugar. That's always going to be the one on the left. And that's the key carbon, right? That's the one with 2 oxygens attached. And then you're going to see where it's linked on the other sugar. So remember, the other sugar has the same sort of numbering system. So we're starting on the right side. So here's the 1. So that's why this carbon is the 4. Okay, so because this one is attached to 1,4, that means it's going to be one of these two, either 1,4 alpha or 1,4 beta. And then the alpha and beta is based on whether the oxygen is slanted pointing up or slanted pointing down. So the one on the left is beta because it's pointed up. And then if you look at the molecule on the right, that would actually be an alpha because it's pointed down. The one on the very bottom here is going to be our 1,6 because you see if we keep numbering the carbon 1, 2, 3, 4, 5, we get to number 6. It's attached to the key carbon here is 1, but then on the other molecule, it's attached to that O carbon, all right? And we made those bonds by combining. We took 2 OHs, basically circled them, and then just combined them into 1 oxygen to make a bond. So if that doesn't sound familiar, go back and watch the class video again and make sure that makes sense to you, all right? Because you might need to either put them together and draw them, like if it's for each bonds, or you'd want to be able to recognize them and make sure you're including the really important parts and not ignoring anything, okay? So the other things that you're going to need to know are about the different disaccharides. So I've shown these up here on the top right. And again, we mentioned this in class, but they're based on the sugars it's made of. So maltose is made of glucose with another glucose. Lactose is made of galactose and glucose. And sucrose is made of fructose and glucose. And all of those are D form. It's just some of them are alpha and some of them are beta. So you can look at that. And then the linkage that you need to know is the 1,4 alpha for maltose, 1,4 beta for lactose, 1,2 alpha beta for sucrose. So if you can look on this picture, there's actually all of these different ones are shown. So make sure you can match maltose, lactose, and sucrose with what you see on this picture. The last thing I want to mention, which I didn't talk about in class, is that disaccharides can also be reducing sugar. So if you look at the bottom here, a reducing sugar it either has a hemiacetal or a hemiketal. Now, if you look at the structure on the top here, that is not a hemi, right? That's an acetal. So where we're making the linkage is always going to be an acetal or in one case a ketal, but generally it's going to be a full acetal. It's not going to be a hemi where the linkage is. However, if you look at the other piece of this disaccharide, it's got another key carbon. So if we look on this side, right? So if you look at this one, is that a hemiacetal or a hemiketal? Yeah, right? That's a hemiacetal. So any of the ones where that key carbon on the right side sugar is a hemi is going to be a reducing sugar. So this one would be 1, this one would be 1, this one would be 1. What about this last one? If you look at the top, obviously that's the part with the bond. If you look at the bottom, that one's also the part with the bond. So if we look at the top, we'd have what kind of functional group? Acetal again, right? If we look at the bottom, what type of functional group do we have there? That's a ketal, right? So it's got 2 carbons, and then that's the key carbon with no OH. So this one is the only one that's not a reducing sugar, and then the other 3 would be. So maltose, lactose are both going to be reducing sugars. This other one wasn't a sugar, but technically if it was put together that way, it could be one. And then a sucrose would be not a reducing sugar. So that's why it's not, because it's closed in and has full acetal, full ketal, instead of having the hemi. The last thing I want to mention about the disaccharides and also what could in theory happen to the polysaccharides is that we can break them apart in a reverse reaction. So putting them together takes an enzyme. Generally taking them apart is a hydrolysis because we're going to chop apart that CO bond, and then we're going to separate them into the separate sugars that go back to having the two OHs, right? So it's like we're adding water because we're adding OHs to the molecule, and that's going to make them back to the regular sugars. So all you have to know about the hydrolysis product is whatever sugar it's made out of, they're just separated. So that's what happens in your digestion, and that's what happens in our lab, and that's just what you get. All right, so that's all we have to say about disaccharides. Let's talk about polysaccharides. So poly, as I'm sure you've heard before, means many. Polysaccharides are just many sugars. So they're actually polymers, which are just molecules that have repeating patterns. And this obviously has a repeating pattern. If you look at the top, you can see this molecule here in a straight line. It's got all these little hexagons. Each of those hexagons is supposed to be like a sugar, right? A sugar in the closed form. So they're all attached here. And as you can see, it goes around this whole thing. And then at the very end, you have the non-reducing side and the reducing side, which we talked about. So this is one reason why polysaccharides are not going to show up as reducing sugars, because even though they do have a reducing end, like 99.9% of the molecule is not reducing. So then that one little reducing end does not do enough reactions to make any good. So that's why you'll see like polymers don't really show up that way. Okay, so there's a couple ways that you can get the polymers to work. Obviously, they can go in a chain like this one at the top, where they're just going from one end to another, and we'll show how those connections look. They're the same linkages we've seen, but we'll just show how that works. And then if you look at the bottom, you've got branches. So like a straight chain polymer would be the top, and then a branched polymer is at the bottom, where it's just coming out like branches on a tree, right? So these are going to behave differently because they're sort of put together differently, and we'll talk about that as well. But what I want to focus on is some examples that you've probably heard of, because I feel like that's the most practical, and then it's kind of interesting because it's stuff you've probably heard of. So I'm sure everyone's heard of cellulose, right? It's in plants. Starch is another thing that's in plants, and then glycogen is something that's in animals. So we're going to talk about those different examples, and we're going to use those to focus on what's important about a polysaccharide. So of course you want to talk about what are these polysaccharides? Where do they show up? What are they made of? Those kinds of questions. And so really we're going to focus on how are they similar and how are they different, because the similarities will help us categorize them and sort of break it down, and then how they're different will help us keep track of which one's which. So one way that they're similar is that they're all made of glucose. So they're not like disaccharides where there's different sugars that could be mixed in. They're literally only glucose. So every single one of these, if you were able to break it apart, you would get glucose. That brings us to the other thing, which is because they're all made of glucose, they can all be hydrolyzed with water and acid or with enzymes. So generally enzymes are the way it's going to be done in vivo, right? So that's the ways that they're similar. How they're different is, first of all, they have different properties, so they act different. And the reason that they act different is because they have different linkages or different connections. So the way they're put together makes them different, then they have different tertiary structures, and then they act different because of that. So that's why their properties are different. These are the things I want you to know. There's actually technically 4 polysaccharides because starch has 2 different types, amylose and amylopectin, and then glycogen and cellulose. So for those 4, for each one you want to know the sugars that make it up, so like glucose and stuff like that, the types of linkages, make sure you could recognize it if I showed you a picture of the structure, tertiary structure, so like alpha helix, beta sheet, and properties and function. And anything you would need to know about that is also in the supplemental text. So right here is a pictorial summary of all the things we're going to talk about. On the left, we've got cellulose. Notice how it's flat. On the top right, we have amylose. It's an alpha helix. On the bottom underneath that is amylopectin. So it's got branches. That's how I can tell. And then on the bottom left is glycogen. But don't worry, we're going to talk about each of these in more detail, so hopefully you'll learn how to recognize them more easily. So before, whenever we were talking about proteins, we saw tertiary structure, right? Tertiary just means that it's a 3D structure of something, and that some things that in the chain that are connected may not be close together can actually be close together depending on the way it's folded. In this case, we're mainly going to be looking at the tertiary structure that's formed by the linkages, because we don't have side chains and things like that. That's not really a choice. So there's no way you can ever get a sugar to fold up like a protein because they don't have the side chains. And they're going to be 100% based on the type of linkage which we've talked about. So first, let's talk about starch. So starch is found in plants, especially starchy vegetables, if you think of potatoes and things like that. Starch is how they store their food, their sugar. So even though we have two different types of polysaccharides, amylose and amylopectin, they're actually both made of alpha D glucose molecules. So if you look down here, it says that. So everything's just made of glucose, and then as the plant needs energy, it just breaks off the sugar. So as we said, the linkages are going to determine the properties. So if we look at amylose, it's going to have 1,4-alpha linkages. If we look at amylopectin, it's going to have 1,4-alpha and 1,6-alpha. That's what's going to make the branches, which we'll talk about in a minute. But also, before we move on, I want to point out that amylose in starch, it's about 20% amylose and about 80% amylopectin. So there's a lot more amylopectin than amylose in starch. Also, another fun fact is that the amount of amylose and amylopectin, the ratio, actually really changes the properties of the plants. So if you think about rice, that'll actually change how sticky it is, how like if it's like goopy and like soft, or if it's like kind of strong, it stays the grain shape. And there's a really interesting video here at the bottom listed that's from Alton Brown from Good Eats. It's kind of an old episode, but it's really cool where he actually talks about amylose and amylopectin, and he's got like a little demo that he does. That's really cool. So you might want to check that out. When you get the slides, you should be able to cut and paste that. So definitely check that out. It's really interesting and fun, and it kind of helps you remember the shapes. So that's cool. And also it's all about food, which makes it even more fun. So let's look a little bit closer about the types of bonds that we're talking about. So for amylose, remember we said it was all 1,4-alpha bonds. So if you look here, here's one of the glycosidic bonds, it says right here. And this is alpha because the O is pointed down, right? Slanty down, which means that if I were going to draw the hydrogens, they'd be up like this, right? And then they're all like that, right? So if I highlight them, there's a 1,4-alpha, there's a 1,4-alpha in that. Now, if I look at the chain, the 1,4-alpha is what's going to make the alpha helix. So the top one here is the amylose, and it's all an alpha helix. That's all it is. So if I look here, it's just connecting each of the sugars together, and then there's hydrogen bonding across the helix, which is causing it to stay like that. Just like we saw in the proteins, that's how you got an alpha helix, is if there's hydrogen bonding in the up and down parts, okay? So that's how I form that. If I look at amylopectin, it's got a mixture, right? So part of it's got the 1,4-alpha bond. So if we look down here, we'll highlight that the same color. That's a 1,4-alpha, right? They're all that. And then if I look here, it's got the same alpha helix. So you can see the alpha helix over here, right? That's part of the 1,4. But it's also got these branches off to the side. So every time there's a branch, that's actually a 1,6-alpha linkage. So whenever I have those is where I'm going to have a branch. And you can see this carbon structure here, and you can see this sugar here has a bond, a 1,4 on the left. It actually has a 1,4 on the right also, and it has another one. So it has like a third bond, unlike these other ones. So that's going up from the 6th carbon. So if we look over here, let's find one of those carbons. And then over here, let's look for those. So if I look right here, here's a 1,6 because it's sticking off to the side. And over here, here's a 1,6 that's sticking off to the side. And here's one. So you can see like anywhere there's a branch, that's a 1,6. And that's what allows amylopectin to actually hold more sugar on it. It doesn't just have to be one long chain. It can actually have branches off to the side. So then understanding that this is a way to store glucose, amylose and amylopectin are basically helping the plant store energy, like we said. That's holding all the glucoses for whenever it needs it, and then it'll break it down. And then we can actually digest starch, right? That's something we can totally digest. So we have an enzyme that helps us break this apart. We don't just use acid. It's called amylase, and it's actually needed to digest this. So starch is kind of complicated for us to digest. So if we just did it in our stomach only, it actually wouldn't get fully broken down. We wouldn't have time. So instead, we have an enzyme in our saliva or our spit that starts the process really early. So it starts to break it down when it's in your mouth, and then as it goes through the esophagus, it can slowly break down more and more until it gets to your stomach, and then a better chance of being digested. And if you can actually experiment with this, if you take a cracker, crackers are very starchy, and if you taste them, when you just chew up a cracker, it tastes like a cracker, right? It tastes kind of bready. It doesn't really taste sweet or anything. But if you take a cracker and chew it like say 10 times and then just keep it in your mouth, in your spit, which sounds gross, but just do it. And maybe wait like, I don't know, 30 seconds, you'll start to taste like a sweeter taste. And that's because it's actually starting to break down into sugar. So you can actually do an experiment yourself with food. And it works with most starchy foods. So generally anything starchy, that process should work as long as it's broken down enough to break open the cell walls or whatever you're trying to get it out of. Okay? So definitely try that at home. That's a fun one. So we still have a few more polysaccharides to go, so I'm going to stop the video here so it doesn't get too long, but make sure that you check out the next video on polysaccharides. In it, I'll talk about glycogen and also cellulose, and we'll wrap up this conversation about carbohydrates. Hope to see you there.